Welcome back. Today we will give you an update on our lecture series, The Bayesian Brain. In fact, another way of putting this is the predictive brain. I'd like to review briefly with you the content of the first three lectures. You may recall that in lecture 1 we outlined the idea that perception is not just a feed-forward acquisition of data from the environment via our senses, such as hearing and seeing and touch and proprioceptive information, but rather the brain uses the incoming information to draw inferences about what's going on in the world, inferences of causality, because it's important to decode the incoming sensory data to make sense in terms of causal contingencies in the world if you want to survive in this world. Now, the idea goes way back to Wilhelm Helmholtz in the 1800s in Germany, and we outlined the idea that perception is matching the sensory information that we receive from second to second with prior information, the so-called priors. And we outlined the utility of these ideas in understanding the glutaminergic model of schizophrenia. Now, in the second lecture, we focused on sensory-motor integration. How is it that you can return a serve of a tennis ball approaching you at something like 80 miles an hour by looking at the racket position of your opponent, the trajectory of the ball, getting your right hand, your forehand in position, and returning the serve. All these things have to be done very rapidly and rely on prior information that is already available in the brain. We also use this idea of making sense of what happens in depression. There is a bidirectional relationship between some medical illnesses and depression. In other words, depression predisposing to some medical conditions, and medical conditions such as diabetes, for example, predisposing to depression. The reason for that may be buried in the brain in terms of a Bayesian inference machine from the insula to the medial frontal cortex, especially the subgenual frontal cortex or Brodmann area 25. And in the third lecture, we talked about that infants use the violations of prior ideas, prior expectations, as they view certain events in the world as a method to draw inferences about what's going on in the world. 12-month-old infants use already valid optimal Bayesian statistics in terms of predicting what's going on in the world. And finally, in lecture 3, we talked about schizophrenia again and the fact that dopamine, a key molecule in the history of schizophrenia, of course, is involved in error detection and the evaluation of stimuli and the comparison of expected reward and received reward. And we made the point that patients with schizotypy or a genetic predisposition to schizophrenia often have an advantage in detecting ambiguous stimuli in the environment by relying more exclusively on the prior information already laid down in the nervous system. So now you may wonder, what is the actual experimental evidence? There were lots of theories proposed. Show us some data that would demonstrate that prior information in fact influences the brain in this kind of predictive coding method that we have talked about. Now, in the first slide here, I introduce you to the paper which will anchor the entire presentation, but we will get to the paper at the very end of this talk. So it's entitled Time Compressed interplay of anticipated events in human primary visual cortex. And it just appeared in May of 2017 in Science Open magazine. Now, in the next slide, I run you through some of the basic ideas we discussed in lectures 3, 4, and 5. Namely, what we perceive with our senses is a combination of top-down knowledge-based prediction and bottom-up incoming sensory information, signals from sensory receptors, for example, the retina. To see a complex world is to deploy world knowledge in the form of a generative model to try to construct the incoming sensory signal for itself using stored probabilistic knowledge about the world. Now, in this predictive processing framework, we hypothesize a multi-area, multi-level system in the brain that perceives the world by trying to predict its own sensory states. Each level at the hierarchy of sensory processing tries to predict the unfolding signal at the level below. Now, the differences between the predictions and the current level, lower level states, are calculated, and only the residual prediction errors are sent forward and sideways in the hierarchy in the cortex to make better predictions and to drive learning. So here then is a schematic. We have sensory input coming in here, for example, to the primary visual cortex V1. And this is the area that the paper that we will discuss, in fact, examines in fine detail. Now, there are some other secondary inputs here, of course, in higher visual areas, the association areas, and two types of neurons are present, namely prior information neurons and error detection neurons. And these neurons in combination in their interplay then determine how much of the incoming sensory signal complies with what already has been laid down in the brain by prior experience and what is old news. And only the relevant error signals, the new news, will be allowed to get forwarded to higher centers of integration. So here then is the classical example of the hollow face illusion. You see here, of course, there is a concavity. The light shines from below. However, if you put some facial features into this cavity, going inside, the brain decodes this as a convexity. In other words, the features of the face coming out at you rather than going into a concave presentation. And that is because the brain has learned with this kind of illumination, this is what you should see. This is the famous hollow face illusion. Now, this means that no matter how hard you try, and you can employ your knowledge of physics and how light works, the brain will override your judgment and will present you with an optical illusion. So this will be the overriding power of prior information, totally abolishing the error signals that are coming in to your brain. All right, so we could say then, in the final analysis, that perception is a controlled hallucination. A controlled hallucination. We already know what we expect to see. We present this image as a hallucination and we correct it by using the error terms that don't agree with the images we already have laid down in our onboard computer. So our brain tries to guess what's out there. To the extent that guesses match or explain away the incoming sensory data, we perceive the world. Now, these are astounding statements because what they imply is that you don't see the world as it is. You see the world as you have seen it in the past. You see the world as you have imagined it or categorized it by accumulated prior knowledge. And only to the extent that you give credence to the error signals produced by the incoming data that don't agree with your model of the world, only to the extent that you give credence can you then correct your perception of the world. Now you can imagine situations where for one reason or another you don't give credence. For example, driving through a foggy landscape and you doubt the visual input coming in because perception is not accurate under these foggy conditions, and you may have to rely more on your knowledge of this particular road rather than the incoming information through your eyes. Now, the error term involves an uncertainty. If we want to guess what's going on in the world, we cannot be totally sure. And as I told you about the foggy conditions on the road, we make an estimate of the uncertainty that we're dealing with in any particular situation, and that is the so-called precision error, or the weights we assign, different weights, different prediction error signals, according to how useful and reliable the information is estimated to be. So precision then acts like a volume control, increasing the postsynaptic gain of selected synapses. Now there's more to say about that, but just to introduce you to the idea of the precision weighting of the error terms. Not all error terms are made from the same mold. Some are more valuable than others, as indicated by how much precision you assign to a particular error term. The dopamine system is critically involved in this operation. So here then is an example. You have sensory input coming in here. You look at a coffee cup. The coffee cup is partially occluded by a structure here, let's say, and the prediction is that this is what you should see. And your visual system can edit out the occlusion that has occurred by an interfering object and tell you, yes, in fact, this is a complete coffee cup and not just some fragment of a coffee cup. So here's then the adjusted hypothesis, and you reconstruct the complete and accurate image of the coffee cup. So that is an example of a error correction term, a Bayesian inference term that has worked well and gives you accurate, updated information of the world. Now let's look how this is implemented. By the way, here are some of the references that you can click on, which will pop up the original PDFs. Now let's look at some ideas as to how this is implemented in the brain, in the cortex, at the neuronal level. So here we have our trusty pyramidal cell in layer 5. And you can see the pyramidal cell really has two aspects to it. The cell body is in layer 5, but the terminal dendrites are in the supragranular layer 1. And the signal coming in is processed in two different ways. Namely, there is a feed-forward system going in here, and there is a feedback system coming here, which is the error correction term that is applied to the sensory signal. So here is kind of the fine structure of this. To the left you see the wiring diagram as evidenced by some retrograde transport of some viruses injected into nuclei in the thalamus. But what we're interested in here is the right side of the panel. And you see again here in layer 5 of the cortex your trusty pyramidal cell. Here you see the dendrites that now can receive the feedback input. you can see that there are two nuclei involved in the thalamus, the non-specific and the specific nucleus. The specific one driving the sensory feed-forward input, arriving in layer 4 and perhaps in layer 5, and feedback system is then rerouted here from the dendrites back to the pyramidal cell. So this then would be the visual cortex V1, and you can see that with this tracing of transported viruses, but also with very powerful and fast MRI, we can dissect these different layers in V1 in the visual cortex. Again, here in the deep layer we have the pyramidal cell, and we have the superficial layer that is driving the feedback. The next slide. You can see what do we need to do? What does the visual cortex need to do in order to detect the presence of a predator that wants to eat you, this tiger here on the left side. And you can see there are external data coming in, fairly sparse, going forward into the visual cortex, and you get feedback from your knowledge of cats in the wild that drive the internal prediction, and when things click together here like so, you make the decision, my God, it's a tiger, and you get out of there. Now, the picture of the tiger needs to be composed, as you know. V1 extracts only very fundamental features of the visual signal, such as orientation, for example. As we go to higher cortices like V4, we decode color, or in V5, we decode motion. So at each layer of the cortex, we have the feed-forward signal and the feedback that's going back to look for error signals, comparing prior notions of movement, prior notions of color features, prior notions of orientation, until we finally, in the infratemporal cortex, arrive at the total shape of the tiger. So this is the kind of hierarchical processing that goes on in the brain, at least according to the model. So now, what is the evidence? Can we do experiments which directly address this notion that there should be events in the visual system that give us a clue or give us evidence that this kind of feedback is actually happening, and there is some kind of influence of higher visual areas onto the initial processing of visual stimuli. And there are many experiments which have addressed this in various ways. This one today I will present to you seems to me intuitively so strikingly clear and obvious that it's very difficult to argue with its persuasive power. So what was done here is subjects were familiarized with flashing stimuli that were presented to them, and from a left to right trajectory, hitting different areas in the retina. And then the idea is, because there is a topographical mapping from V1 to the retina and from the retina to V1, whereby the structure, where the spatial orientation, the spatial features of the stimulus are preserved. They're directly transmitted from the retina to V1. So if a stimulus goes from left to right on your retina, you can find that trajectory in your V1 area reproduced faithfully. And so human observers then were exposed to a dot that rapidly moved across the screen from left to right or right to left for a 4-minute period while maintaining fixation. So after this exposure period then, occasionally only the starting point. It's called the pre-play condition, or the endpoint, the no pre-play condition of the sequence was flashed onto the retina, omitting all the intervening dots. So the reasoning then is that if V1 is involved in the prediction of anticipated events, cortical activity during the cue-triggered preplay should resemble the activity during stimulus presentation at retinotopically defined locations while preserving the temporal order of the dot sequence. That's a mouthful. Next slide will show you how this is done. So here's the retina, and you have the retinotopic mapping. The different quadrants of the retina are faithfully produced in V1, and you have some cortical magnification here of the retinal signal. So here then is the task. You can see that the stimuli are presented like this. The dot is flashed and it moves from left to right. You fixate your eye on the cross in the middle, such that you don't pay attention to the dots, but the dots will go from left to right on your retinal surface. Now, in the control experiment or preplay, only the first dot is flashed. And in the no preplay control condition, only the last dot is flashed, the end dot. So the prediction then is, the system learns that there is a left to right sequence of dots. The brain has now learned to anticipate when this dot is played, it expects these other dots to occur as well. That would be a proof of the hypothesis that the system anticipates the next stimulus. And how does it do that? Well, because it has acquired a prior, a dynamic prior, that for 4 minutes it has learned that a dot will appear, followed rapidly by several other dots moving from left to right. Let's see what happens. And here's what happens. So this is the experiment, the stimulation. And here then is what happens during preplay. You only fire the first dot, and yet you see the brain responding. There's no stimulus coming here, but the brain believes there ought to be a stimulus now. And there is a system that tells V1 to hallucinate and to produce the missing stimuli, because we now have learned when the dot appears here, several other dots will follow very rapidly. Now, if you only play the last dot at the wrong position, in other words, here, there is no preplay, because the system has not learned that a dot on the right-hand side is followed by additional dots going from right to left. Now, it doesn't get any easier than this. This is such a powerful experiment. What it shows is that the system, the brain, the visual system in the brain has learned to anticipate incoming signals, that even if they don't come in, the beginning of the cue of the sequence will trigger the entire avalanche of events that the brain has anticipated will occur. Here's another way of showing this. So this is the location of the dot, and you can see the different locations expressed in different colors here. And in the preplay here, you can see there is a good separation of the first dot, second, third, fourth, and so on. Whereas in the no preplay condition, only the last dot here shows up in the response. Now here, some statistics are now applied to this data to sharpen it up and to reduce the noise that is inherent in this response. And now it becomes even more clear that whether you attend to the stimulus, this is the attended visual field, or you put a distracting event in, you get pre-play events happening when you play the left dot. The right dot here, the normal condition will not generate this response. So in the next slide, are there any consequences? Does this mean anything for the performance of the visual system? Well, yes, it does. Interestingly, the replay of the dots, in other words, the hallucinated dots, are time compressed. In other words, they happen faster than the initial presentation of the sequence of dots. So you can say here, this is the peak time, and peak time here is shorter than the peak time of the original stimulation training. In other words, the brain is not only replaying the sequence, but it's replaying it in a fast-forward mode, allowing you to take action faster than you could have otherwise if you only replayed it either at a delayed speed or the normal acquisition speed. The other question is, what about accuracy? Does this training, the pre-play acquisition training, does it do anything in terms of the accuracy of the response? So what was done here is to put some arbitrary delays into playing the last dot. And you can see that the detection accuracy is improved in the pre-play condition. In other words, the brain is more accurate in predicting not only the sequence, but it's also more accurate in the local prediction as to where the signal has occurred on the retina, correcting for the error introduced by the experimenter here in this case. So accuracy and time are both improved by this pre-play phenomenon in V1 of the retina. Now, what is unusual about this experiment is that all this happening in V1. We used to think that things happen V2, V3 in the more higher integrative areas, that's where manipulations occur. But it's as though at the entry point of signals from the retina to the brain, already feedback loops are in place that powerfully examine the incoming information and make statistical predictions about the information, such that when you have a surprise event in a sequence leaving out an expected item in a sequence, all hell breaks loose in V1, and the brain will make sure that you form a facsimile of the information that you expected would happen. Now, there are other experiments of a similar kind that also are now in a cumulative way supporting the notion of a feedback loop that encounters the incoming forward volley of sensory information. At the anatomical level, if you look at the nerve connections, the fibers in the brain, it turns out that the feed-forward fibers are perhaps less than 40% of the connectivity from the lower sensory to the higher integrated sensory cortices. It seems as though perhaps more than 50 or 60% of the connectivity is downward or feedback from higher cortical areas into lower cortical areas, suggesting that the feedback condition or the examination of the incoming signal by information already present at higher areas is extremely important to how information is processed in the brain. So this was an update then on the Bayesian brain, or the predictive brain, the predictive processing framework. This framework has been applied to many other areas of brain function, such as sensory motor integration, to the analysis of moods and affect, for example, to the formation of beliefs and many other areas. And we will expand on those in the future some more, because what this promises to be, perhaps, is an integrative model of not only sensory processing or perception, but perhaps also sensory motor integration, the formation of belief, and perhaps other cognitive functions that can all be accomplished in this particular Bayesian predictive processing and error minimization framework. Thank you very much for your attention, and we'll see you soon again for another talk at Behavioral Health 2000.